Let's go, take a seat, take out your review packet. Let's go, Alex. Phones away. Get going, thank you.
You're welcome.
Joe. On your phone. I just said phones away. Alright, I'm putting Sit down. Where's your review packet?
You guys said that 500. Did you hear the attitude in that? Yeah.
Hey, let's go. Let's go. I will raise my voice whenever I want to. No.
Can you please?
Oh shoot, it's on my computer.
But he's very important. You last year 24. Guys, everyone get kicked out today. Make them smile the whole period.
I'm not doing what I want. Oh, there it is.
You can help them get sold from this line.
Yeah, incredible game. Alright. Incredible. So, wow. How's this review packet coming?
It's going first page.
Almost.
I got two
Eight.
Listen,
I'm here.
guys, just listen. This is going to take you quite a while.
Yeah, thank you.
So you need to get started on this.
Well, it's not due till Tuesday, correct?
Is it due on Tuesday or is it Why would you do this morning?
All right. I cried. So we worked on Punnett squares. All right. But chapter, let's see, chapter 8 was your worst test.
Thank you.
Yeah, I think the average was like a 64.
That means I got all averages. I got 67.
Not that bad. Good for you guys.
I literally think I got 40%.
Generation of winners right here. Okay. So what is? Hey, listen. I don't want to beat up on a student the last day of class. I don't want to do it.
Just kidding.
For this whole year, you've been building me up, and I might crack.
What's that song you build me up, you grab me down?
I got pop ones in me right now.
What's that song? You build me up, you break me down.
I got
an
86 on what, Joe, what is the structure of a DNA molecule?
It's like Katy Perry. A double helix.
Really? Nope. What is the DNA molecule made of? Guys, I will kick you out on the last day of class. Okay? Your advisor will get a very nice email saying how mean you were in class, mean and disrespectful, and how you made me cry a little bit inside. And they probably won't take that lightly. So what is the structure of a DNA molecule? You've had your hand up. Can you answer that?
Yeah.
AJ. Nope. So it is a double helix, but what's the structure? Okay, so sorry. Yes, that is correct.
Yeah.
That is correct. I'm wrong. I'm sorry. But what is a DNA molecule made up of? Oh, Billy.
Lots of stuff. They're made of nucleotides.
Okay.
You want me to keep on going?
Chapter 8. Well, it's made up of more than that.
I know. Whoa! Calm down over there. Also made up of well, like there's the central dogma, but that's like a process that goes.
Watch this going over to there, champ.
Okay. There's phosphate group.
There is a phosphate group.
How do you display sugars? You want to know about
No, there are sugars.
Yeah. Ribose
Yes.
sugars, deoxy.
Yep.
Ribose sugar.
Wait, just go to the bottom.
What the?
Just for that, I'm going to take you left me hanging. So now
Yeah,
you're
that
hanging all over that.
Did I have a photo of it on my computer?
Okay, so yes, nucleotide. Okay, what are the 4 different nucleotides for DNA? A and T.
A, B,
Yes.
C, G.
Thank you.
Are we gonna need a
Okay, I'll pause for a second. AJ, what's your question? Is there a review? So there's enrichment, yeah.
Wait, is
Actually,
that Saturday? No.
Oh, no, we got
No, Monday afternoon.
Awards.
Isn't it Monday afternoon?
There's afternoon study hall, isn't there?
Monday afternoon.
There's enrichment from 7 to 8.
You sound like you went
But
to
it's not. But it's not for my class, right?
No, Monday afternoon is for biology.
Oh, yeah. I don't think so, actually.
It's for English.
Yeah.
What?
Okay, so our review is going to be Monday afternoon during the afternoon study hall, and Monday night during the Monday night in Richmond. All right. So we'll have 2 different reviews. I don't have a senior exam, so it'll just be bio and you guys.
Oh,
Wait, forensics doesn't have this?
oh
I'll take forensics as a junior.
So mine.
Okay.
Why is it junior? It is a senior. You have some-
What is the central dogma?
It's a
Jack.
3-step process.
Jack.
I just said that. I just talked
Jack.
about that.
It's a 3-step process that goes from duplication or replication to
What is replicated?
DNA is replicated,
Yeah.
and then it's transcribed
Into.
into RNA, and the RNA is translated to a protein.
Boom. All right. Be familiar with the central dogma. All right, because that'll help you out. If you just know the central dogma, that'll help you
What's the
with
final?
the rest of chapter 8.
What's the final product for central dogma? Like, is it
Protein.
okay?
All right. Also, know where. So besides of like know what replication, transcription, translation is, know where they occur. All right. So
Wow.
where does replication occur?
Inside the
Right. Yeah.
so it's out into the.
Where does transcription occur?
Wow. In the ribosomes. Or no, that's the other one.
Insane in the membrane. What insane?
Are you going to be guilt-toeing it in a second?
I actually said he said that.
Membrane of the nucleus or whatever.
Transcription. Joe,
Uh,
where does transcription occur?
I think so,
Yeah.
Why is terminated?
No, wait.
Henry.
Where does transcription occur? On the central dogma.
No, isn't it like
Boy, is it, yeah. Here, I can tell you.
Yeah.
In the body. Oh my gosh, the net, that's a great answer. No. Oh, whoa, wait, so.
Stay on this question.
nucleus?
Yes.
Wait, they both have the nucleus, and then it goes down.
That's what
Oh,
I'm saying, but that's not
Because
even
what is
simple.
what is transcription doing?
Making-
Making. Right. And where is DNA located?
Nucleus.
Right. So from transcription, what's the product? What's one of the products? The main one
Of what?
of transcription.
Oh,
RNA.
mRNA.
mRNA.
So from transcription, we get RNA. The RNA is then going to travel where?
To the ribosome.
Outside the nucleus to the
Ribosomes.
good. Where what is going to occur?
Translation.
I think more of the stuff.
He majored in biology.
Do you have a question or you just taking a selfie? Like I don't know what's going on.
There's an Eiffel Tower on my screen.
He's using photo, but
I'm drinking water from a. No, I actually need this because then I'm not gonna get anything else right because my biology final exam thing is
Your
on.
screen's gonna crack.
It's gonna break it and you're gonna owe me
No.
$2,000.
actually 1.2 grams.
Because everyone else saw you throw it against the floor.
They saw you smash it.
No, we didn't.
What is the purpose of a carrier? That's such a weird question.
Oh, I know. It carries like energy.
Purpose of carrying.
Yeah. Oh no, it's when you have a disease.
Like when you have a recessive.
Yeah.
But you don't express it.
Yeah, but
So the
what's the
purpose, I
purpose
guess,
of
would
it?
just be to
To know more about it.
not express that trait, which could be harmful. Okay, going down this list again for chapter 8, we have a couple polymerases. Those are enzymes that are involved in those different processes. You should review those videos that I posted, all right, on Veracross about replication, transcription, translation. Those are all from YouTube. They are great sources. You can even go on YouTube and look at more. All right, they're all pretty good at giving a nice visual about the process. All right, anticodon and codon.
What the hell? Probably should have been writing these down.
So if I draw up an example
Anticodon is the opposite of a codon.
like this,
It's what matches up to a codon.
Should we hit the running the down?
Yes.
You know what?
We definitely should have.
You're still writing. That's a shame, Steve, because I don't have
You sure
my
that
computer
was-
or my phone.
Alright,
What?
He shared that with us.
He told me I.
I'm doing it online too.
Wait, did you share us with us?
Did you share it something? Oh, can I do it?
I got it. Never mind. He did.
alright.
Thank God.
So if I give you something like this,
What is this? The term? Ever?
I'll explain a few of them as I go down. Let's say we want to go to DNA. Joe, what's this first letter going to be? Okay, Joe. Get Chipotle out of your head. Okay, it's not happening with me tonight.
Can I guarantee
Chipotle
it?
is not going to happen. So when we're making up a new DNA sequence from this mRNA
Oh, you want me to do that for you?
sequence, what's this
T.
first letter going to be? D?
T.
D?
Yo, it's watch like
T.
Boy, you're wrong. It's A.
Damn it.
Got this.
What a
Alright, here, let me go again. Give me another try.
Redemption.
G.
G.
H? Yeah.
Yeah,
Brad.
Jokester.
Next one.
Yes, you know you.
G. A.
Henry.
A. U.
He said, hey.
T.
Oh.
Joe.
What?
Stop talking, Joe.
I'm
Jackson next.
U.
Oh.
Wrong.
T.
T.
Mr. Fork, you have a question.
I got it said T and you
Joe.
heard me.
C.
Alex.
Ooh. Ooh.
Look at the
C.
one.
G.
G? Yeah. G.
G. G. No, wait.
G.
G.
If you think about it, that's an
J.
interesting word.
Alright. So remember, DNA base pairing rules is what? A to what?
A to
And G to
C.
good.
Okay, so
RNA base pairing rules, raise your hand if you're going to talk, is A to Cam. Me?
No.
What is me?
What? The letter.
I'm a letter?
Yeah.
Your letter, all right.
Wait, is that a top?
And G to what?
This is our last biology class.
It is.
So no,
Which
you
one
can
is that
question.
I'm crying every time. It's important. You've been waiting for it. It's here.
Let's
So
go.
tRNA.
We have a question.
It's like my birthday come early. tRNA.
I have a question.
When's your birthday?
I said.
Next
Wait, so
Hang on.
You got a pun.
mRNA and tRNA, both of them, T goes to you. Wait, not eDNA?
Okay, so A goes to you.
Yeah, well, because it
There's
would
no
be
T in here though.
Okay. And then to find the amino acid, use your tRNA.
No, you use your mRNA.
Okay,
And I'll explain that a little better next. Alright, but making tRNA, what do we got? Tyler, first one.
A.
Go take a water break.
You sure?
Yes. See you, Tyler.
It's not
I
Tyler.
get it. I get
Yeah,
it.
you just said C Tyler.
No. That's I asked Tyler for the answer. He thought.
Ah.
Yeah.
Comedy.
Then what? Okay, good then. AJ, what do we got next? I know I am.
Hey. Shots fired.
Next one.
Next one. A.
A. Bert. Is it E?
No, I just didn't.
You. You.
I said that's soldier boy.
Jack.
C.
El Diablo.
Sell it.
Oh, Jay.
How did you hear
Yeah.
my nickname? That was like my Spanish class thing.
Where is that from?
Yo.
Okay,
You heard from Talladega Nights and you thought it was fighting chicken.
Tyler, finish this up.
You gotta keep the highest number. Whoa.
Nice.
Someone just ripped.
No,
You
that's
went
my
over.
shirt.
It was, wait, Mr. Forky. But it gives you the mRNA, so why would you go and
Wrong.
do the
No,
DNA?
wrong, Mr.
It's
Forky.
escaping issue.
You farted.
Stop talking. Billy, what's up?
If it already gave you the mRNA, like why would you use the DNA and cRNA?
Because what if I ask you,
Oh, I saw you.
what was the DNA strand that the mRNA came from?
And then you just write what you have up there.
Alright, oh,
Well,
Alright.
if you did it,
So what is a codon? Jackson.
It's like
Is that
part
right?
of the DNA that like tells the thing to stop copying it.
no, that's a stop. That's a stop code on.
It's a start code.
Oh,
It's stop code
It lets you know what part to
Stop
stop.
code.
Okay, so you're talking about start codon, but just in general, what's
It's just like a thing on
Oh.
the DNA strand on the nucleotides or whatever that like tell the
2040.
thing to like start
I'm
copying.
hearing a lot of things.
Like,
Okay,
I
so
know.
all a codon is, yeah, no, take another one. But I need to do my packet.
You're still thirsty.
Okay, so all a codon is a series of 3 nucleotides that are going to code for an amino acid.
Yes, that's what
Jackson.
I still don't know the purpose of sex.
Alright, there's a start codon that starts the translation process, and there's a stop codon which ends that process. Right, Henry?
The start codon that
I
starts the process.
didn't ask you anything.
Wait, Mr.
I
Forky.
have a question.
What's on your computer?
Technically, you did ask me, just nothing.
Which was 3
Notes.
Why don't
nucleotides.
we
On your computer?
What is the purpose of a sex-linked gene?
Sweet,
What is the sex link gene?
it's a gene that's
Fetal
linked.
Brad,
allele
take a break.
related to
No, I have a
organs.
question.
That's incorrect.
Yeah, see?
Mr. Forky,
It's a gene relating to
what is the purpose of a sex-linked
Hang on, I'm still on this
Are
one.
you gonna give us a
Wait,
no, I had to go pee anyway.
Mr. Forky.
I know.
I am not listening to anyone because they're not raising their hands.
How do you know you turned around?
Oh!
Wait, what is that?
Joe.
So, codon is 3 nucleotides that code amino acid.
So, you use those things.
So, you use the mRNA strand to code for those amino acids. So that's what we're going to get our codon.
So you use those.
Alright, let's just say this isn't actually what it is, but we don't have the chart in front of us, right?
Would
So
you
let's
give
just
us
say
a
that that codes for serine, this, tyrosine, this, glycine. Let's just say. Hang on. Now, those are codons. What are these called?
amino acids? Oh, they're called.
Didn't I tell you to take a break?
Yeah, I know.
Take a break.
Take a break. I love you. I got this. You don't want to
Spam. Let's hear it.
Anti-codon.
Anti-code. Hey, those are good stuff.
What? Why are you getting compliments on
Yeah,
this?
but Ross
Other
is-
things are-
I
Anti-code.
would've gotten 50 compliments then.
You wear just tips with ankles on.
Alright.
Wear
Anti-codon.
anything with ankles.
Alright. Now, the reason that you don't use tRNA
What is it, anti-codon?
when doing this, you don't use the tRNA codons to code for those amino acids because they are they're on the same molecule. Okay? So, you right in that corner of the room? So, this one tRNA molecule,
Can you just share that to me? I don't want to know this down.
So I'll just do it. You give it to
No,
me.
this is how you wrote it all down, not the answer.
Oh.
Hey, Mr. Fork.
so that's tRNA.
Make it so
Joe, take another break.
I've raised my hand for like 5 minutes.
What'd you call me?
Can you answer my question?
No.
I've
So
never
this
Mr.
one
Spoonie.
tRNA
Can I take a walk with Joe?
has that anticodon strand as well as the amino acid attached to
I'm a
it.
guess then.
Alright? So, when you have that mRNA strand, it's going to be UCC. Okay, you're going to have that tRNA molecule
Noodles.
No problem.
tRNA noodles.
out here with that amino acid attached to it. So this is why you're going to use this mRNA strand, alright, to translate into amino acids, because this anticodon is going to match up, okay, with the mRNA. It's going to drop off the amino acid and start building that amino acid chain, which we're going to be calling a what? What do we call an amino acid chain? Amino acids.
Protein.
Protein.
Wait, why do you use tRNA? Amino acids are monomers of
Did you honestly just ask me why you use the tRNA after I just explained it?
What's tRNA? Yeah, you told me to walk out.
I don't even know what tRNA is. Is it working?
Okay, because this is the tRNA molecule up here, and the amino acid is it's attached to this tRNA molecule, as well as the tRNA strand. So we're going to look at this, the mRNA, to see what tRNA molecule we need to bring down here.
Why did he use UCC instead of UAG and GCC?
Do you know how long this is going to
Okay,
take?
that's my first one. Then next would be
Now, why are you doing all that? Why wouldn't you just have to get the amino acid out?
next would be that. And then there's another tRNA molecule that codes for that.
And then it goes down to that UAG.
And then it's going to be going down here.
Now, do you have to diagram all that stuff?
No, just know the general process. You're not going to have to draw.
So the answer